Hi everyone, Microbehunter here again, and yes, I think I'm live now. Okay, it always takes me a couple of seconds to get everything sorted out here at the beginning. Yeah, today I'd like to talk a little bit about diatoms. And here we already have a microscope slide for diatoms. I'm also going to show you how I'm going to use the centrifuge here in the corner. You're able to see a centrifuge. And I've seen that a lot of people all over the world already started to comment. And I would like to say, of course, a hello all over the world. And like every week, I would like to quickly ask you if the sound is fine, because sometimes it's a little bit too loud, sometimes it's not, it's a little too soft. People have problems understanding me, but I hope that everything works out. Yeah. So yeah, there are people again. All sound is good. Very good. Okay. Yeah. So today I'm going to look at a few diatoms now. First of all, diatoms are quite common. I think many of you probably have already seen them before. And what I would like to do today. Oh, and I forgot it. Okay. Maybe later on I'd like to do a little bit of oil immersion microscopy of diatoms as well. Just see that I forgot my immersion oil over there at the cupboard. I might have to jump up and get it later on. Okay. Yeah. So what we're going to start off right away a little bit with a slide that you see right now here under the microscope. I might actually move the other camera away from the centrifuge to the microscope. Again, it's a commercially prepared microscope slide, and it just says here diatoms. Okay, it doesn't say how they were prepared. It doesn't say from where the diatoms are from. There are basically a whole range of round diatoms, and this basically is an indicator that we're probably looking at fragments of so-called marine diatoms. So basically from the ocean, because very commonly they are round. Okay, that's basically what I suppose. Now, when you actually go in a little bit higher here, okay, you can see that they really look quite nice. Okay, so usually what I do in the first 5 minutes or so, I'm not really going to dive in right away, but I'm simply going to wait until more and more people start to join. Because according to the statistics of YouTube, it takes about 5 minutes, 10 minutes for people to join in as to see the notification that the live stream is active. And during this time, I usually read the comments just want to say hello to everyone, especially when people are telling the community from where they are from. Okay. Hello to everyone. Looking forward to an interesting session. Well, I hope it's going to be interesting. I have to tell you, if you joined the session last week where I put some of my face mites under the microscope, I think the face mite one is going to be hard and very difficult to beat. Yeah, because diatoms are not quite as active. Some of them do glide around and move around. They're pretty important, extremely important, as a matter of fact, because they produce a significant amount of oxygen in the Earth's atmosphere, and they look beautiful as well, but maybe they're not quite as scary and as the face mites. Face mites are also not scary, but some people don't like them. So I'm going to, therefore, every now and then, you probably already know that I'm going to quickly interrupt every now and then my talking, and then read some of the comments. And if you're new to the live stream, yes, please do comment. I have to change this here to live chat at the top, okay? And if you basically also have questions that are not directly related to the topic, but they're somehow microscopy related, I might also answer them, okay? So, so hello from Vietnam, from Argentina, okay, from Mecklenburg in Germany, yeah, so that is from Hungary, New Jersey, United States, okay. Yeah, USA again. Yeah, so that's really nice. Vienna, Austria. So I've been thinking of cutting a little bit of my dog's fur to make a slider of the dog's fur. I've also done this already before. So what I'm going to do today is the following. India, okay. Yeah, I would rather have diatoms instead of mites. Well, yes, I'm going to start off by showing you a video, which some of you might have already seen, because some weeks ago I made a video in the very same channel where I put I set up coffee machine under the microscope. And this is basically the video because I found some interesting diatoms. And in the water container here, it has not been cleaned for quite some time. Basically there was a biofilm there. And essentially what I did is I put this biofilm under the microscope, quite simple. And this is where I saw those diatoms here. And basically what they are algae. I also made a nice video here. And then somebody commented on the video, and I just want to talk a little bit about that because I found this kind of interesting. When you turn on the arrow over here, these are the individual diatoms, of course. You see that there is a green chloroplast in the middle here, and those things on the side here, apparently these are fat droplets for storage. Somebody who commented in the video actually said that they must be kind of fat droplets, in the green part in the center here, that's the chloroplast. Yeah, so those diatoms are actually algae, and what they have is they have a shell made out of silica. Silica is the chemical which is like glass. Okay, and some of those shells, the frustrules as they're called, are quite elaborate and nice looking, and that's why we're going to have a look at a few. Okay, that's not a diatom. That's looks like an amoeba or something. But I just wanted to show you here a little bit the biofilm, and then for comparison, I want to show you something else. Okay, you see that there is quite a substantial layer here, green layer. And what I would like to do now is I'm going to go back here again, and I want to show you something else. Okay, look at this. You cannot see it because of the green screen, so I have to put it down here. This is a water bottle. Okay, and this has been in use now every day for a year. And let's have a look if there is also a biofilm of diatoms in there. And not surprisingly, there is none. The bottle, even though it has been in use and for a year now, there is no biofilm in here. And the question is, why is that? Why are there no diatoms growing in here, but in the coffee machine there is? Okay, so that's the fundamental question. And the answer is very clear. There is a reason why there is this dark cover around it, because this, of course, prevents light from actually reaching the bottle. And the diatoms, because they're photosynthetic, they need light to grow, right? And for this reason, it's important to always keep this cover on here, not only to kind of protect the bottle from breaking in case you drop it, but actually it's a light protection to prevent the growth of a biofilm inside here, right? So I just wanted to clarify this a little bit, is so that if you have somewhere some standing water for a longer period of time, and if there's light able to approach it, then of course it's quite likely that diatoms and other algae are going to grow. Okay, so comments are flooding in like always, which is very nice. Okay, I might not be able to respond to all of those, and sometimes there's a little bit of talk going on between the people of the community. That's perfectly fine. Okay, but so there is and sometimes there are questions. How about fresh urine? I heard it's sterile. Is that really the case? Indeed, urine is sterile, should be sterile, and only becomes contaminated during the process of urination. And then, of course, if you have urine outside, then of course it's also going to be contaminated. Being organic, it's of course also a food source for bacteria, and this basically means that urine which is standing outside for several days will spoil, because bacteria will use the organic material. Urea is, as a matter of fact, organic. Will use this as a food source, and that means that they will grow. Okay. So my dog is sleeping. Okay. Slightly unrelated, but I just caught a yellow sac spider in my flat in Vienna. It's the third one this year. At least it's interesting to look at under the microscope. And what I recommend is the following. Of course, insects and spiders are very good to look at using a stereo microscope. If you don't have a stereo microscope, use a flashlight and use the low power 4x objective. Okay. Can they survive without light? I suppose you're referring to the diatoms, and the answer is of course, for a short time they might survive without light. That's the normal part. I mean, you've got the night, day and night, obviously, but for a longer time period, I don't think that they're able to survive. Okay, because they are photosynthetic organisms, diatoms, and they need light to actually make their own food. And to grow. Yeah. Does anyone have tips on how to find hydra? You gotta go to a pond or lake and get some sediment, some solid material. And I do not want to keep you waiting right now. And I'm going to start off by preparing the first slide. And you're going to be a little bit surprised, because I bought myself 400 grams of diatoms. 400 grams. Look, Here it is. Okay. Ooh, and yeah, the spots here are because of the green screen that I have. Yeah, it's called Diatomaceous earth. I bought it online over Amazon. It says here made in the United States, but actually it's, I don't know, it's a different language. I think it's Polish or so. So it was made in the USA, it says here. So I suppose it was harvested in the United States. And it is sold, I would never eat it, but it's sold as a food supplement. Here it is. I don't know why you would want to eat that. Okay. But this diatomaceous earth are basically fossilized diatoms. And this has also commercial use. So for example, apparently I'm not an expert in this, but dynamite, the explosive dynamite actually contains this substance here because it has a very high surface area. And so the nitroglycerin, the explosive material, is kind of absorbed to the diatomaceous earth, which is very porous. And this kind of stabilizes it. I've also heard that it's used in some tooth pastes to kind of as an abrasive, so to say. Yeah, and this one is sold to be eaten, but honestly, I don't know what it's good for. I wouldn't try it either because silica, and that's basically what diatomaceous earth is and what diatoms are. The shells, the diatom shells is silica. Some forms of silica are actually not so good if you inhale it. Okay, it can cause a condition called silicosis. You're pretty bad for the lungs. But I heard again that the diatomaceous earth is not as problematic. But again, I wouldn't risk it. So I bought this because as a demonstration material. Okay, so what I'm going to do now is I'm going to simply open it up, and I would like to make a microscope slide, and we're going to have a look at some of this diatomaceous earth under the microscope. Plain and simple. So what I need is I'm going to open it first. Maybe I'm going to make it a little bit larger so that you can actually see the contents here. Yeah, this is how it looks like. It's just white powder. That's it. Yeah, it looks like almost like flour. And you need a very small amount here. And what I'm going to do is I'm simply going to dissolve it in a little bit of water and put it on my microscope slide, and that's pretty much it. So I'm going to take. You're not able to see this from the side here. It's here, a little sample of this powder. Let's put it here into the dish. Okay, and let's close it. And let's have a look at it under the microscope after I added a little bit of water. So maybe I'm going to bend down the camera again a little bit more here. Do you see this a little bit better? And yeah, I always have this, by the way, I always, because I don't have running water where I am, that's why I bought myself this little water bottle. It's kind of blowing air now. Okay, yeah, and it starts to dissolve. Well, not dissolve really. It's suspended, so to say. Yeah, and and then I think I'm just going to take a pipette. Yeah, and I use this pipette here. You already know it, and then. Usually can properly mix it by drawing it up and down a little bit here. And then let's put a small sample, a very small sample here on the microscope slide. Up the small sample, way too much. Because I want to go to high magnification, and therefore I must make sure that it's really very flat. Where are my cover glasses? Okay. Cover glass goes on top. I sometimes have to check this every now and then because those cover glasses I put under the dishwasher. No, I didn't. Not on the dishwasher directly, but I added some dishwashing powder and cleaned it that way. It's fine. Here we go. Any excess liquid should be removed, and that is basically trial number 1. Simply some diatomaceous earth under the microscope, and let's have a look. Yeah, I have to remove, of course, the commercial diatom slide here. It's a little bit too bright now. So let's put the diatomaceous earth here under the microscope, and I have to focus now, of course. Okay. Yep, that's what I expected. And not very spectacular, it might appear, but we have to go up with the magnification to see a little bit more. So that's 20, it's 40 times. Let's refocus a little bit. because these are fragments of no. And we still have to go up a little bit more, because if you're a little bit patient, you are able to sometimes see that the structure is quite nice. For example, this one over here, the square one over here, almost in the center. And let's go up a little bit here. There is the movement that you see over there. I don't think that there cannot be bacteria. This must be kind of Brownian motion of whatever particles are there. Okay. And let's focus a little bit here. Yeah. And yeah, those square ones I found quite frequently here. These are the shells of some fossilized diatoms. Yeah. Very often fragments. You have to be, occasionally you see some nice structures, but because these are basically fossilized forms, I've rarely found a complete one. Okay, but you can already see, let me get the arrow back. Okay, you can already see over here, yeah, they have, yeah, the many of these are a square, and they do have a shape, but then they're kind of broken apart. Yeah. Yeah. So this is basically, yeah, because of the high surface area of this, so that's silica, right? Because of the high surface area of this diatomaceous earth, it's been, yeah, it has a range of industrial uses as well. Okay, so let's yeah. If you look at this more often, you're able to see that there are always some kind of recurring structures, like bent ones. Yeah, the square ones are quite common. Maybe the density here is not so high. I've actually yeah, square ones, for example. Yeah. Yeah, I did make some months ago, when I bought this box of Diatomaceous Earth first. I made also a video which is simply called Diatomaceous Earth, which you're also able to find here in this channel. But you see that, yeah, also over here. Yeah. So again, maybe a little bit disappointing, because we are kind of, yeah, here at the top, because you're kind of used to diatoms being nicely shaped. But again, here, because this is fossilized, and I don't know how old they are, quite old. Yeah, and because it's been in the ground and compressed, and yeah, and for this reason the pressure must have also destroyed them a little bit. But because you see that some of them, the particles are rather small. I mean, these are nano-sized particles here, right? For this reason, I would be rather a little bit. I wouldn't want to eat those. Yeah. Here again, you see exactly those square-shaped ones. These are quite. These are some kind of recurring structures that you're able to see. Yeah. Yeah. So let me go a little bit up with the, I can also change a little bit the polarization around. Yeah. So, wow, lots of comments. I'm a little bit concerned that I might not be able to answer all of them. Why no diatoms in your bottle? Maybe you flush the bottle once in a while. That was kind of the question. This refers back to the bottle, this invisible bottle that you're not able to see because the color is green. The green screen. Yeah. So the reason why there are no diatoms in here is it's not because you're flushing it, because what they do is they form a biofilm and they won't be flushed out. They will actually start to grow on the inside of the bottle. And I can imagine, hypothesis, but I mean, what is the glass made of? Yeah. Okay. Yeah, it's basically the same material as the shell of the diatoms. So it's quite well possible that maybe it's even using the material of the glass to make those diatom shells, and because there's no light able to reach it, therefore they cannot grow. Yeah, so they're still talking about the dog and dog fur. Okay, and where did you get the sample that we're looking at? Amazon, really. I simply looked at Diatomaceous Earth in Amazon, and this is basically what I got. Yeah. And yeah, way too much material. This is the smallest one I could get. You need only very small material, obviously. But yeah, what I might do is I might go up a little bit with the concentration, because I am a little bit disappointed with the amount that we see here. So let's do this again. Yeah, let's add a little bit more. Okay, let's add a little bit more of this here. Let me add a little bit more. Yeah. You see that it dissolves or it becomes suspended quite easily. This might not be a little bit overdone as well. Okay. Again, a small drop here. Let's try it again. Maybe they're a little bit more. Yeah. So in other words, I just wanted to show this to you that these are essentially fossilized diatoms and that they do have a significant commercial use as well. I think as abrasives and for industrial use and of course also for making dynamite, interestingly enough. Yeah, so again, this is a little too, yeah, here, this is a little bit better here. A little bit more. Always go up with light intensity. Yep, that's better. Yeah. Yes, okay, I think this looks a little bit nicer. You get the idea. Okay, now you see a more different shapes, also round ones. Yep, that's actually the thing, that the way I wanted to show it to you. So there was simply not enough sample material on my first try. Yeah, now it would be kind of interesting to find out how old they actually are. Yeah, but you can also see now that there are also round ones around. Okay, so that's basically, yeah, these are just the diatom shells. Obviously, they are not alive anymore. These are just basically like the skeletons, so to say. Chloroplasts are not present anymore. And yeah, for this reason, they, of course, yeah, are just fossils, so to say, dead, obviously. So I'm going to go quickly again into the comments. Okay, this was a commercially permanent slide. Yeah, the first one that I showed you, I'm a little bit late maybe with some of the comments. That the first slide that I showed you with the nice round ones, that was a commercial one. And I mean, it's labeled in German. What does it say? Diatoms. That's it. Yeah, it doesn't say anything else. Yeah. Yeah. So, and if you are really into diatoms a lot, there are a whole bunch of traditional diatom slides. Some of them are quite expensive because back in the 19th century, some hobby microscopists had a interesting pastime. What they've done is they've actually picked out individual diatoms and arranged them in nice patterns to be observed on a microscope slide. And how do you actually pick out individual diatoms? What they've done is they've taken an eyelash. If you put an eyelash here, under the microscope, and they glued it to a little handle, and because an eyelash is very pointed, and because of the natural oil and fat that's on the eyelash, you're able to pick up individual diatoms and place them in a nice pattern on a microscope slide. So this was actually a very common pastime at that time. And apparently those traditional microscope slides obviously are collector's items and not so cheap, apparently. Now, I don't own those, but if you Google online a little bit about traditional diatom slides, you're able to find a few beautiful ones. And if you see the diatoms kind of arranged in a round pattern, then you know that they have been placed by people most likely using their eyelashes. Sorry, I meant to say house, Dornfinger, not okay. God, is okay. I kind of missed now a little bit the context here. Okay, hello from Massachusetts. So I have to go back a little bit and read the previous comments because dynamite. Yes, dynamite, the explosive. Yeah, it actually contains originally. Contains also diatomaceous earth because the explosive substance, which was a liquid, actually were kind of stuck to the diatoms, which have a very large surface area, and this kind of stabilized it. Yeah. Okay. Yeah. I like the beautiful glass shells. Okay. So yeah, so what we're going to do now is now that we have looked at a whole bunch of dead diatom slides, let's try to look at a few live ones. And what I would like to do now is I simply want to also show you a little bit how I do that with my how do you call this, the centrifuge. I do have a centrifuge here as well. okay, which I'm going to use. Maybe some of you have already seen me use it before. I think it's not necessary to use a centrifuge, because what you can do is you can simply wait for the diatoms to settle down for a couple of days. Yeah, but centrifuge also works, okay? So we're gonna try the following now. I have over here, you know this already because I show it to you quite frequently. I have over here a jar with algae growing in here. Again, it looks a little bit weird now because of the green screen, right? But there are not so many. I don't see a biofilm growing on the side here, but I know that there are plenty of diatoms in the filamentous algae. And what I'm going to do now is I want to get them out, and I want to concentrate them using a centrifuge, right? About the centrifuge, I got this over eBay, new. It was ridiculously cheap. It was around, I don't know, 30 to 40 euros, $30 to $40. It does its job, really. It's not a great one, but honestly, it's good. It's good enough for my purposes. It's a little bit bulky, I would say. I mean, there are smaller ones, but essentially, yeah, plastic, totally analog, but that's fine. And then I'm also bought myself those yeah, tubes here, centrifuging tubes. They're disposable, but I reuse them. And inside here, and I don't know, you have to look in here a little bit. I'm going to point up here. Yeah, you've got the rotor, okay, and there are those plastic insets here. And what I will do later on is I will yeah, put those tubes here with the diatom solution in here, and I need to add a balancing tube on the other side here as well. Okay, it's got to have the same mass, otherwise it's going to vibrate. So that's really important. So I will use a scale to weigh it, right? And then I'm going to spin it for a couple of minutes. Over here, there's a timer and there's a speed control. And then I can put a lid on top here. Yeah, it's going to make a loud noise. And then I'm going to just, yeah, look at the centrifuged sample, and then I'm going to put it under the microscope. So that's a fast way of actually concentrating those. So let me just, yeah, so that you see it again. And how do you do that? One way is the following, that you simply take a little bit of water. Oh, where's my water? Where's my water bottle? I lost my water bottle. No, that's the alcohol bottle. And I put it somewhere and I forgot where I put it. Can you believe this? I just had it a minute ago. And where? Just a second. This is not possible. My desk is not that large, but there's so many things standing around that I essentially don't know where I placed it. Could it be that I... This is kind of embarrassing. Just a second. I must have placed it somewhere unconsciously. And I don't know where it is. This is kind of weird. Okay. Otherwise, just a second. Maybe it fell down somewhere. This never happened to me before. This is the alcohol bottle that I have. I've got another one over here with alcohol. I just had it here. Just a second. It's diatom. Hey guys, I put it somewhere unconsciously, and I forgot where I put it. It's kind of funny. Okay. But sometimes it's like this, that you simply have the things in front of you and you don't see them anymore, simply because you're so no, it doesn't matter. What I'll do is the following. I'll take a little bit of some liquid from here. But behind the monitor I put it. Look at this. Here it is. It was almost hidden behind the monitor where I had some free space. Okay. Here it is. Okay, so I'm gonna fill this up with a little bit of water. So now I have to remember where I put it, here, right in front of the monitor. So and I'll take now some tweezers, and I need some scissors. And I'm going to take now some of the algae out of here. I'm going to move this a little bit up. So, and yeah, I don't know if you're able to see this. I'm gonna hold it up here. Yeah. So lots of algae, and here you, yeah. And I'm going to simply cut this off. And I'm going to take this now, and I'm simply going to rinse it in here. Okay, of course it's going to slowly turn green, but I do not want the filamentous algae. I just want to, yeah, get out whatever is yeah, suspended in here. This means not only diatoms, but maybe a whole bunch of other living things as well. Probably also some other single-celled algae as well. Okay. So I'm going to simply put it out. Again, we can see that everything starts to turn a little bit green already. I'm going to try it with the next one. Okay. So let's take this out. All of the larger parts I'm also going to take out. Okay. Ah, there's some solid material in here as well. Okay. Of course, it's possible maybe to put it directly under the microscope as well, but I simply want to show you how this whole thing works now with centrifuge. And I'm going to start to simply fill it into one of those tubes that I have here. And I'm going to do this a very easy and cheap way by simply pouring it in like this. Okay. This is about what 12. It says here around 12 milliliters, 12 mils. I can see or you can also see it's slightly greenish. So evidently there is something here, which is good. And now we need to do something pretty important. We need to balance this. So I'm going to use another one here, simply, yeah, as a stand. I put it in here. I also put the cap on here, and yeah, it's now to yeah. I don't know if you're able to see it. It's now to 0. You're not able to see it. It says 0, 0, 0, 0. Then I will now remove this. Again, I will close it off. And I will put it somewhere where I don't lose it. I put this one in here. And now I've got minus 11 grams, 11.08. Maybe you're not able to see this, but I have to now add a sufficient water until it's also back to 0. Okay, minus 3.75. Okay. Up, yes, exactly, 0.000. Okay, so this basically now means that both of these tubes now, yeah, both of these tubes now have the same mass, and this basically means that I can now put it into the centrifuge, and yeah, one over here. You're not able to see this again, so I put one in here, the other one in here. Yeah, right across each other. Lid goes on top. Okay, and yeah, I've always set it to the maximum, and this over here is the timer, and now it's going to be noisy. No, that's just a second. No, that's the timer. Okay, and that is that's the speed here. Yeah, I'm not going all the way up. Okay. Okay, this is how it looks like. Yeah, and while it's running, I'm going to. I don't know if you're able. I don't know if you're able to hear me properly because of the noise. But yeah, I'll simply have a look now again at a couple of comments here. So the question is now, does each country have the same diatom species, or can other varieties be found? Both yes and yes. I mean, of course, there are certain species that can appear all over the world, but it's not really a question of country, but on the local situation. I mean, different ponds, different bodies of water have, of course, different diatoms, but I'm quite sure that some of them might be quite similar. So I mean, there are marine diatoms that can be mostly found in salt water. Then there are freshwater diatoms. depending on the temperature, on the pH. Yeah, everything's possible. And of course, sometimes you might be able to find the same species. Yeah. So hello from New Zealand. I saw a video on Journey to the Microcosmos that says one study found that diatoms and flagellates in extended periods of darkness. Okay, I don't know about that. I have to look at this. So maybe that they're able to survive extended periods of darkness. Okay, what is Brownian motion? Yeah, Brownian motion is, let's go back here, is the random movement of small particles. Okay, so let's have a look here. Not that may. It depends a little bit if we've got enough water or not, but sometimes, now that's because of the vibration of the movement that you see now. Sometimes it's because of the vibration of the table, but sometimes you will see small particles shake and move around, and that is because the thermal activity and the water molecules bumping into the particles, and so it's a random motion. That's called Brownian motion. The movement that you see right now, a little bit, is because the table is vibrating, okay, because of the centrifuge. Okay, like ancient fossils, yes, Diatomaceous earth actually are ancient fossils. Okay, so yeah, like one of the shapes. But yeah, they talk about the shape of the diatoms. Could you show how it looks like in dark field? Yeah, I can. Problem. Dark field for me only works with the 4x objective, and it doesn't look nice because it's not magnifying enough. But dark field actually would be a nice thing, but this is not gonna. This doesn't look nice in my case. Okay. In my case, it doesn't look nice because I don't have. Sufficiently high magnification. Okay, now that would be dark field. Okay, so all of the bright spots that you see are individual diatoms. Forget it. Yeah, that's simply has to do with the setting of my microscope. Yeah, so not that dark field in my case, not a very good option. No microscope is perfect. But if you can attach a dark field filter, it would actually be quite nice. Okay. Okay. Yeah. Where am I? Just a second. Okay. Yeah, I love diatoms in dark field and DIC. Would it make sense to put this suspended flower in a centrifuge to get the bigger, more complete diatoms together, or would it destroy them even more? If you are okay, if you I think that it will. Let's put it this way: it is indeed correct that you have to be careful not to spin them too fast, otherwise the G-forces might break apart some parts. However, I don't think that in this case it would make a huge difference because many of them are already broken apart. The problem with centrifuging is that it depends more on the density. So this means that the smaller, the larger ones will not separate if they have the same density. Okay, so that's a little bit the issue. Yeah, it would be nice to kind of separate the larger ones out first, but if they have the same density, they might not separate. Yeah. So what my magnification is this? This one is now, I'm using, this is the maximum that I have right now. It is 60 times. So I'm using the 60 times objective. Okay. So I personally think that diatoms are the coolest things to look at. Yep. Okay. Yes, it was 60 times. So I'm skipping down again. Can I see diatoms in lake water? Yes, you can, but not in the water itself, but collect some sediment, some sand, some material from the bottom, and or scratch off some biofilm from rocks, okay, or from some plants. Anyone you want to buy an autoclave? Somebody wants to sell an autoclave? Okay. Okay, some general comments. I scroll down. And then I think I'm just going to stop. Could we use diatomaceous earth to make glass? I don't know, honestly. Are you able to melt it down? Maybe, I don't know. Okay. Hello, I would like to know how to record a time lapse. Basically, what you can do if you, I think this might now be enough already. And I hope that you were at least able to understand me a little bit. Okay. If you want to record a time lapse, what you do is you simply record a movie and then speed it up in your video editing program. So let's have a look now. Let's see if we were lucky. Okay. And we were lucky. Okay, look what I got here. Okay, this is just this one over here is the balance, the water, and look, this one is the one that's centrifuged, and you look, it looks much more clear, and look what we have here in the bottom. This is called the pellet. Yeah, this is basically where all of the diatoms and whatever algae I found collected. And now what I have to do now is I have to now carefully decant the water. Depending on how solid the pellet is, you either can carefully pour it out or you have to use a pipette to take it out. I'm just going to use a pipette, and because it's the safest way of actually not destroying the pellet, because sometimes accidentally you can also flush out the pellet this way by simply pouring it off. It depends a little bit on the nature of the substance. Sometimes some pellets are very fragile and they flush out immediately. Others are quite solid. And so you can already see that this liquid here is extremely clear. So that all of the green stuff and I expect not only to see diatoms here, or hope to expect, but maybe many other things as well. And carefully I leave a small drop in here. Okay. You know what? I'm going to put it down again a little bit more, and I have to refocus here. Where is this? Okay, here. Yeah. So there's a small drop of water, and then usually what you can do is you can either tap it. See? Yeah, it's already resuspending. It's very dark green. Or you can use a pipette and you can pull it up and down. That's also a possibility. But I'm almost a little bit concerned that this might be even too dense. Yeah. So look, that's how it looks like. Okay, so a slide. Okay, we save the rest here, and put a lid on top, and we take a cover glass. Again, I have to check it's reasonably clean. And honestly, I have not tried this today in the afternoon yet. So for me, it's also going to be a little bit of an unexpected thing here. And let's have a look. Let's take out the other previous slide, and let's put in this one over here. So yeah, I mean, I do see some stuff here. Mostly green algae. I don't know if there are plenty of diatoms present as well or not. Yeah, a whole bunch of single-celled green algae. Maybe mostly not diatoms, I think. But yeah. The water is moving because it's not quite even. Okay, well, these are honestly, some time ago when I tried this, I saw more diatoms, but these are some other algae. Well, but I still have another sample to show you. Here's one. Oh, it's kind of disappointing. But where is the arrow? I don't know if you're able to see it. those irregular shaped structures that you see most likely are diatom shells. Yeah, well, yeah, not only most likely, this is almost certainly. Yeah, but that's actually a pretty tiny one, considering the fact that. Yeah, here's one, here is one. But honestly, it could be that maybe some diatoms are actually also arranged in a sequence like this, but I'm not so sure about this one here. That looks also could be. I've seen nicer ones, honestly. Yeah, but that's a diatom shell, definitely. Yeah, but actually the idea was also to show you a little bit how centrifuging works in case you have not seen this before. But these are all of those round things that you see here. These are not diatoms. These are other single-celled algae. But the nice diatom shells we don't have now. Here's another one. Okay, here's one. Here's one. And you see that there is quite a bit of liquid streaming still as it spreads beneath the cover glass. There are several ones over here. Here's one. Here's one. And in order now to actually concentrate those diatom shells, there is a method that I actually will try out as well, but it's a little bit dangerous, okay? So because the diatom shells are made of silica, which is inorganic, okay? Therefore, you are able to remove all of the other organic material by mixing this sample with hydrogen peroxide, concentrated H2O2, very aggressive stuff, and then you basically heat it up on a water bath. Now, what is the H2O2 going to do? The H2O2 is so aggressive that it's going to oxidize everything that's organic. Basically, it's going to convert it to carbon dioxide. And what is left over? Left over, you have calcium carbonate. Some shells of calcium carbonate. And you have left over the silica shells of the diatoms. The calcium carbonate can then be removed by adding acid, like vinegar, for example, concentrated vinegar. This will remove the calcium carbonate shells or sand particles that are made of calcium carbonate. And then at the end, then you want to probably centrifuge it again. And then you have the pure diatom shells. Okay, so this is basically a method of concentrating those is by removing everything here that you see here with hydrogen peroxide. And then by removing any calcium carbonate that is present of shells with acid. And then you have your pure diatom shells left over. So there are some other organisms also seem to have survived the ordeal of centrifuging. If you centrifuge it too much, of course, you're going to kill many of those because the G-forces are too high. Yeah. But I think the problem that we have here is that many of the diatoms are actually covered up by the much larger single-celled algae that you see here. Okay, so that's a little bit one of the issues. But with the hydrogen peroxide, I do want to try this out. Here and some nice diatoms over here on the side. But you got to be careful with that stuff because if you use here a nice large diatom as well. Okay. But what I'm seeing is that most of those diatoms actually don't seem to be alive anymore because they, how do you say, they're not green. Yeah? Because normally they should be green. Yeah. So what I'll do now is again a couple of comments and then I'll look at a couple of other samples, searching for some of those. Okay. I was able to get a video footage of diatoms 1,800 times. Okay. Okay, hi, I would like to know how to record time lapse. Okay, that's basically what I said is, yeah. So there's some communication going on. 1,800 magnification. Can you share the link? Okay. Could you post a link for the video or wait for us to watch it? Okay, this is for someone else. Yeah, you have a reason for table. You, if you want to buy yourself a centrifuge, I don't think it's absolutely necessary, but it's nice to have. And yeah, this is a fairly large one. There are smaller ones as well, that so-called that are used in molecular biology, and I don't know if I have one of those containers. In here, they don't use those large tubes, but the small plastic tubes. Unfortunately, I don't have one here. Okay. As a matter of fact, I think I might have one. No, I don't. There are other centrifuges around that use those small plastic tubes. Okay, Eppendorf tubes, as they're called. Yeah, it's a question of preference, I would say. Those here have the advantage that you're able to centrifuge a larger quantity. Okay. So I'm reading some questions. Refining uranium at home. Oh, I tell you. Yeah, you need centrifuges as well to purify uranium. Yeah. So, okay, no, not so many questions so far. Okay. I used to use an ultracentrifuge when I worked in in research. We used ultracentrifuges in the lab to concentrate DNA. Yeah. Okay, the people talking about kombucha. Kombucha also under the microscope looks quite nice. So you know what I'm going to do now? Let's move on a little bit. I want to show you another sample. And this one is 1 year old. And what I would like to do here is I would like to be very careful. I'm going to take a new tip. I'm going to trash this one over here. Okay, because this sample here, I hope that it is still alive, does contain also some small nice diatoms, and I collected this sample here. There is it's salt water, marine water with sand in northern Germany. Basically, in northern Germany, there not only in northern Germany, it goes all the way to the Netherlands, Belgium, yeah, all the way down. There is it's a natural, very nice area where there are very large tides. So it means you can walk out into the when there's low tide, you can walk very large distances. And I took a small sample along from there, I also made a video once about this, and the diatoms survived for a couple of months even. So I have not been checking this now, and I'm going to simply take you can see here a small sample of sand water. Okay, and let's see how this looks like and if they are still alive. Okay, because there were lots of diatoms present. The problem with that sample is every time when you have a thicker sample with sand grains, of course, you cannot go up with the magnification quite as high. But this is also a very nice specimen for time lapse, because you are able to see then the movement of, provided that we're lucky enough, you're actually also able to see the movement of some of the diatoms, provided that we're able to, that we're patient enough. Okay? Because some of them actually do move, but very slowly. And if you look carefully, go again a little bit here, yeah, then you're able to see that, yeah, all of those things that you see, many of them are diatoms. Okay, for example, here also in the corner, yeah, and look, some of them are gliding. So they actually survived almost a year, yeah, in this jar. Yeah. And yeah, what they do is they happily live doing photosynthesis, and sometimes, and I made a video, of course I did, you're actually also able to see this on the time lapse, the massive movements, because sometimes they're actually attached to sand grains, and then they even push the sand grains around. Yeah. Especially in time lapse, when you speed up the video, this is quite fascinating to observe. Yeah. So let's go up with the magnification yet more. Yes, here we go. Okay. So some diatoms are able to glide and to move around. So this little jar with some marine sand is still quite alive. Always make sure if you want to keep water samples for a longer time, always make sure that, and if you close it off to prevent evaporation, you can do that, but always make sure that there is plenty of air in the jar. So otherwise it might become anaerobic and then the whole thing is going to start rotting and then it's not so nice anymore. Yeah, everything's going to die off. Yeah? But this one is the sand sample from northern Germany. Of course, we can also have a look at some microplastics. This is another project that I have. Yeah, that essentially very often you're actually also able to see pollutants and microplastics and other impurities in the sample. Yeah. Yeah. So again, a couple of comments here. Okay. Yeah, people talk about kombucha. Okay, some people curious about the 1,800 times said. Okay, the centrifuge is inaudible to us, so no need to speak louder. Oh, that's good, because actually it's quite yeah, it's maybe because of the directional microphone. Huh? Okay. For some reason my audio has some back noise. I hope maybe this was because of the centrifuge. Yeah. Yes. Yeah, I made the yes some time ago. I actually because some people requested, how can you make your own centrifuge? It is possible to improvise a centrifuge. It's a little bit dangerous, maybe, but what you can do is you can take an electric drill, one that or you know it's used to also maybe an electric screwdriver, and then if you mount a round disc like a CD, for example, or a round wooden disc, and then if you connect those tubes to the disc and have it spin, then this actually also works. Yeah, but you got to be careful if this kind of comes loose, then you've got parts flying around. But it is possible as well. Yeah. So again, a couple of questions. What magnification do you need to see diatoms? Not a lot. Honestly, the diatoms here are not very spectacular. I have to admit, I've seen actually nicer ones, larger ones, but honestly, if, you can see already the basic shape starting the 10 times objective, so 100 times and higher. Yeah. As a matter of fact, if you are able to get yourself a nicely prepared diatom slides only of diatom shells, then this sometimes is also used as to test the optics of the microscope, because sometimes some structures of the diatoms are so fine, and if your microscope optics are able to resolve that, then you can actually make a microscope optic comparison using using diatoms. So this is actually also commonly used to test resolution. Yeah. I recently started to concentrate suspended organisms using the tea leaf effect, which is very satisfying results. That's also very nice. The tea leaf effect is if you stir something in a dish or so, and as the water spins, then you're going to see that sometimes the particles start to collect in the center of the dish, of the cup. It's called the tea leaf effect, because like tea leaves that spin, then basically will collect in the center, and that's also one possible way of concentrating specimens. Yeah. Yeah. So noise of the centrifuge. My microscope is having problems that it casts a shadow when I close the condenser, and when I close the condenser fully, it's still much brighter than normal. Try to adjust the height of the condenser as well. So maybe it has to do something with the condenser height. Okay. So, okay. Yeah, a lot of beautiful cells. Okay. So, I have to tell you, however, that I have seen some water samples where the diatoms were actually much larger and much nicer. It is a little bit a question of luck as well. But yeah, let's have just a short look here. yeah. Maybe what happens if I take the polarization away, then you've got that's now a regular bright field, and sometimes you're able to see the colors a little bit better. Let's turn it up like this, because yeah, the green color of the diatoms chloroplast makes things makes it appear a little bit easier sometimes. And sometimes there's a movement going on, obviously. Yeah. So I guess we just put it back again here. So I want to have a look at another one in this case, but I have not tested that yet. So I have to see if, what time is it? Okay, it's 1 hour. So we still have around half an hour. I just want to try another one here. And this one, I don't know. Okay. So I'm not very optimistic about this, but I just want to see how well this actually worked, because some time ago I found a puddle after rain in a forest, and it was old enough for algae to grow in there. So I'm kind of hoping that yeah, in the sediment down here, there you can see some filamentous algae. I hope that maybe there are some diatoms in there as well. If not, it's also fine. We can look at some of the other algae that are in here. But I simply would like to put this directly under the microscope. Okay, so I have to again exchange the tip here. Okay, you can actually also exchange the tip by pressing this one here. And let's have another look here. And this one is now indeed a little bit going to be a little bit unprepared, because I really don't know what to expect. And I just realized that this might not be long enough to actually reach the bottom here. I'm going to use a pipette, a disposable pipette here, to take a sample from the very bottom. Okay, so there's some solid material. Ooh, a lot. Some solid material has collected. Okay. Let's see if we're able to find anything interesting in here. And cover glass, of course. And yeah, let's have a look here. And there's still too much liquid here. Gotta be careful, because it can happen that if you use too much water, then of course you're going to dip the microscope objective into the water. Not always a huge problem, just have to clean it again. But while doing a live stream, of course, I cannot clean it so easily. But that is now basically the sediment, and it does look green, right? Ah, no, paramecium. Okay, okay, paramecium. At least we got that. So that's a paramecium. And if there are not many diatoms, then this basically means that the puddle, so to say, simply did not have enough, simply wasn't old enough for them to form. But occasionally you're still able to find them, and maybe this is simply not large enough. And yeah. Okay, so this is a sign that we actually don't have anything interesting here. But there is movement going on over here. What could this be? A gliding diatom? Yes, it is. Most likely a gliding diatom. Go up a little bit with the magnification here again. Yes, definitely. Yeah, they look quite nice, especially also on the time lapse, obviously. So let's quickly go up again 60 times. Here we go. Okay. You can see, of course, the center of the diatom is green, because it's the chloroplast, the photosynthesis, and it's moving around. Actually, this one is moving quite a bit. So Are those ones undergoing mitosis? Well, I don't know which one you're talking about right now, but yeah, diatoms are able to, of course, have to undergo mitosis as well. Okay. Well, I have some blue-green algae in my blood. Really? You shouldn't. And if you do get some blue-green algae into your blood, then your immune system's gonna take care of this. Okay. Okay. I collect samples at a local salt marsh and always get tons of diatoms, at least 6 to 7 species. Yep. I've been, yeah. So, permanent slides of diatoms. If you want to make a permanent slides of diatoms, then of course you want to probably make a permanent slide using only the shells, the silica shells of the diatoms, because yeah, they look actually quite nice. Yeah? You have to remove everything else then, and that's basically what that is mentioned before. Is you have to use hydrogen peroxide, or you yeah, are somehow able to pick out the individual diatoms and arrange them on the slide. Huh? So how do the diatoms move? Samples from a turtle pond are full of diatoms moving. That's the movement of diatoms. It's interesting. I did read something about this. It's a gliding movement. I'm also not quite familiar with the mechanism, but yeah, it is. Yeah, they do not have cilia that push them forward, right? But they actually somehow on the surface they're able to glide along a surface. Yeah. Yeah. Okay, I'm just reading the comments. I would like to have a minute of your time if possible. Sure, you can ask a question. Yeah. We have this in our water and I'm sick. Well, let's put it this way. It depends not all that. I mean, there might be poisonous diatoms also around that might be problematic, like they're poisonous algae. Some blue-green algae are problematic. But I mean, as you've seen with my coffee machine, this was not a problem. Also because they yeah, were sticking to the wall of the container. Yeah. Yeah. So I don't know much about microbiology, but I really enjoy your content. Yep. I mean, that's the whole idea, is a little bit of this channel and this live stream. It's more about nature exploration than about any advanced science. I deliberately want to, I mean, I'm a microbiologist myself, and especially in the field of microbiology, it can become very technical and chemical very quickly. But I deliberately, yeah, want to place a strong focus on more on nature observation, because I think that's sometimes a little bit also forgotten, you know, simply the curiosity, and yeah, and that's also one of the reasons why I sometimes simply take a sample and I put it under the microscope in a live stream, and I sometimes don't even know what to expect myself, because it kind of it's the more realistic way of actually going about, yeah, and exploring the environment. Yeah. Do the hard diatom shells affect cell division in any interesting ways? Well, it's a little bit like this, that cell division, what I read, works like this, that there is essentially a larger shell growing inside a smaller one, so like a box inside a box. Yeah. And so that's somehow how they're growing. So when a new cell forms, then basically a larger cell that encapsulates the other cell half will kind of form over it. Yeah. So this is somehow how they are able to divide. Yeah? I see some diatoms that look like sticks, yeah, and not only that, there are of course also round diatoms, but the really nice ones, unfortunately, that I have also found occasionally, I'm unfortunately not able to see here. But I've seen some very large, beautiful diatoms. Maybe what is this one over here? Yeah, that's the paramecium again. Okay, yeah, I've made a lot of paramecium videos already before. That's the paramecium, and And what you normally would probably do now is I would take a little bit of this sample and add a little bit of food, yeah, maybe a tiny amount of milk or milk powder, yeah, or a wheat grain, and then this is going to feed the Paramecia are going to also reproduce. Okay, so what do you see over here? This is the I have always a problem pronouncing it, Spirogyra. That is not a diatom, but it's a filamentous algae, and also very common, and is characteristic because it has this spiral-shaped chloroplast. Okay, yeah, so that's why it's called Spirogyra. It looks like a sine wave here, but actually it goes like a spiral because it's a three-dimensional structure. As a matter of fact, you can see this. Yeah, that now that when you focus, you can see that now this here is in focus, and this part is blurry. And then if you focus it differently, then this one becomes in focus, and the other one over here is blurry. This actually shows that this is not in one plane, but it's actually spiraling. Yeah. Okay, but that's not a diatom. Okay, this is the filamentous algae. Sometimes it might be useful to go down a little bit with the magnification to, yeah, because you simply get a wider view of what's there. Do you know if the jar of diatomaceous earth was separated from marine sand like this, and how, or are they farmed somehow? To my knowledge, they are farmed, and it's not, they don't take the diatomaceous earth out of the ocean, but from land, which once was ocean. So I know that in Germany there are some regions that have large amounts of diatomaceous earth as well. And it's simply on land, so it's kind of simply farmed or kind of like simply harvested, so to say, and taken out of the ground. So again, here Spirogyra, not so many. Okay, I don't know about this one here on the top. What this is? Yeah, so you see that. That look at this, isn't this? This is also that's cool. How many of you know already what this could be? I know, this funny star-shaped thing here. What is this? Oh, of course. Let me see right in the comments. It's not a heliozoan. Not a heliozoan. It's an amoeba. Okay. Heliozoans are much more rigid, and what you are able to see here, look here, those extensions that you see, these are so-called the pseudopodia, and they're able to change their shape. So you see over here on the left side here, something, flowing out. So that's an amoeba here. Okay, heliozoans are much more rigid, and I would say a little bit more regular. Okay, so if you find one of those guys, then I recommend that you do a time lapse, obviously, because they like to move, and if you speed it up, then you can see it much better. I think it looks quite nice, especially on the time lapse. Yeah, maybe we're able to observe how it engulfs something. Huh? Yeah, so you see that in a water sample, usually there are a lot of interesting things to observe. So we've basically gone from trying to search for diatoms to actually finding an amoeba. Yeah, but you know what? Diatoms can also be found in our tap water. The question is, could be, but then I would say then the tap water, I mean, in low quantities, certainly, but then you have to ask yourself, where does it come from? Where does it come from? Because usually the piping, if it's dark on the inside, then they cannot grow in there because there is no light. And if you find diatoms in the tap water, then they must have originated from the source, I would say. Okay. One of the band members from Sparrow was also a student of, okay, yeah. Is there a reason why you used your old sample from northern Germany in seashore? Well, yeah, it's the sample that the reason why I used this here is because I visited northern Germany last year, and I simply took this along as a little souvenir, and I simply wanted to see how long I'm able to keep it. And generally, I find salt water to be much more easily can be kept much more easily. I feel than fresh water, and yeah, and I simply wanted to see how yeah, how much life there is between the sand grains, and that's the reason why I kind of kept it. Yeah? So this is a little bit out of personal interest and curiosity. Yeah? A while ago, I read that Diatoms exclude a thin film of mucilage from the Rafi and glide along. Seems it's still not well described in either way. Yeah, so it's kind of some kind of a some kind of a sticky substance, but how it actually uses it to move, I also don't know. Yeah. Yeah. So should I make a permanent slide? Well, actually, I wanted to make a permanent slide of some diatomaceous earth. With the reason why diatomaceous earth because it's completely dry, and because I would like to compare a little bit the refractive index. Because when making the diatom slides, the refractive index of the mounting medium is extremely important. And for this reason, I want to, yeah, indeed, try to use this PVA glue, and also the Uprol mounting medium, which is actually more used for yeah, for insects and so on. I would like to try both to make a permanent mount, and then yeah, I would like to then compare maybe next week if one of them actually works better to get the details of the diatoms yeah, out. Yeah? Because if the refractive index is not correct, then there are plenty of paramecia here, then you might not be able to see all of the details. So that's a little bit also the experiment. Yeah? So, okay, so what do we see here? We learn here is that there are, okay, maybe that's a diatom here as well, but not a big one. Not a big one. So, but that's of course also a diatom here, right? Yeah, so maybe I'm gonna do that. But it's 1:17 now, and we're still around a quarter of an hour. So let's try to do that. It's going to be a little bit of a challenge now because I need to clean and always wipe this, and let's try to make a permanent mount of this diatomaceous earth. And here we put it here, and maybe I'm just going to collect some of the powder in this dish here. Because every time when you test something out, it's always a good idea to use standards. And here I know it's pretty pure. And this means that because it's pure, I do not have any interference from any other organic material. So, and what I would like to do is I would like to now try this PVA glue, which I've already recommended before. This is, yeah, simply water-based glue. And I added a little bit of water to it to dilute it down a little bit. And, yeah, and I'm going to simply apply a drop here on the microscope slide. And I'm going to add, of course, a little bit of this diatomaceous earth directly into it. And then, of course, it's a question of whether it actually sinks in. This is now the thing where I need just a second, maybe something like toothpicks. Do I have toothpicks? Unfortunately, I don't. But maybe I find something else that I could use. Yeah, I don't have anything here. So what I'll do is I'm simply going to use my pointed tweezers here, and then I need to wipe it to kind of spread it apart a little bit. Otherwise, it might be too difficult to see. And yeah, we add a cover glass and wipe it again. And maybe you want to press a little bit to spread everything. And again, the reason why I'm trying this with two different mounting media is because I would like to see the effect of the refractive index. But we can check it right now. Always be very careful when you do that, because there is of course glue spilling out here on the side. So for this reason, I do not want to use a too high over magnification. Otherwise, there's the danger of me dipping the microscope optics into the mounting medium. And of course, air bubbles. And yeah, this is how it looks like. Not so nice compared to what we've seen before. But I'm simply going to let it dry a little bit, and then once dry, I mean, we can see the tiny diatoms here. Yeah. I cannot go up with the magnification. I don't want to risk it. Okay. But some air bubbles here. But I'll prepare it now, and then maybe next week, when everything's been dried, maybe we're able to compare a little bit which mounting medium actually results in the best contrast. Because if the refractive index of the mounting medium is the same as the refractive index of the otherwise transparent diatom shells, then you're not able to see them properly. So this is the reason why the choice of the correct mounting medium is actually quite important. Okay, so this is now basically the one with the glue. Okay, so let me put this somewhere. And the next one would now be I would like to now use U-Pearl, which is otherwise my favorite, or one of my favorite mounting media, because it's a very nice general purpose one, with a disadvantage that it actually dries very slowly. So usually it takes around a month to 6 weeks to dry. Okay. Ooh. Okay. Here we go. Unfortunately, I got bubbles. Not good. Okay. Unfortunately, bubbles. Yeah, but they might actually pop with time. Yeah. So let's see a little bit to what extent this mixes. I don't want to use the tweezers here. Maybe I'll find something else. I've got a little, look at this here, little wire. To break apart the clumps a little bit. And again, we add a cover glass. We press to make sure that everything spreads nicely. Let's have another look. We can already see that. Yeah, there's of course, some bubbles. Yep, and this is basically how it looks here. You see that the mounting medium obviously is still liquid, yeah. But there seem to be no clumps forming, so this basically means that the mounting medium and the diatoms seem to be compatible. Otherwise, they would kind of clump together. You see there are these air bubbles. We're just kind of now trying to find their way out. Okay, but I think that's going to be fine. Yeah? So in other words, yeah, what I'm going to do is I'm going to simply allow both of them to dry. Okay, and then we can do a side-by-side comparison, which one actually looks better. Okay, because as I mentioned before, the correct refractive index is very important for getting the best image quality, especially with transparent objects like those diatoms. Okay, so let me quickly go through some of the comments again. I'm going to start at the very bottom. Do you ever use Canada balsam as a mounting medium? No, for a very simple reason. That Canada balsam requires the specimen to be completely dry, and you need to work with xylene, and I don't want to do that because xylene, or also known as xylol, is not so healthy. And Canada balsam was commonly used previously, and has a very good, apparently very good long-term storage ability, especially if the slides are properly made. But it has become a little bit difficult to obtain, because you have to prepare the specimen properly. It's basically somewhat a more advanced mounting medium. Yeah. Okay. Okay. Just can you show microplastics? I'll do a separate video of those. If you take water samples, for example, and you put some sediment in there, very often you can find tiny fibers, which are very often microplastic fibers from clothing. So synthetic fibers from clothing can be found in many places. Actually, it's quite sad where you can find them. Yeah. Do you have a local place where you can consistently gather radiolarians or forums? Okay. Radiolarians and forums, I don't have a place. They can be found in some marine samples. I do have, if you're interested, I do have some permanent slides here, but I don't have a place where I could find those. Yeah, because I'm landlocked in Central Europe. I don't have access to a marine environment. Yeah, seems like lately you have shown all your microscopic samples exclusively by DIC. In fact, your DIC pictures look great, and for me, this is a very strong incentive to try out this technique. Okay. Short information. I'll put in concerning DIC. So what you see here, let's go back to this sample again. Is if you look at this sample again, it looks a little bit three-dimensional. It looks almost like the cells are kind of pointing outwards, right? That's the method is called DIC, and it uses polarized light and a couple of fancy prisms and so on. However, you can imitate this technique almost free of charge. And even today, I got an email from someone who managed to do that. You do a combination of Reinberg illumination and oblique illumination. So what you do is you basically make sure that you show this to you here. You use a filter that looks like this. I mean, it's 3D printed, but essentially you can make this yourself by simply taking some clear plastic and coloring the center. This will give you a blue background. And then don't put it into the center, but you kind of, in the filter holder, you swing it out halfway so that the light strikes the specimen from one side. And then you also get a very similar effect, practically free of charge. So this is called oblique illumination. And if you now use it not with a black one, but one with basically is kind of blue, then you also get this blue background. Actually, the picture that I got sent today was surprising. I mean, I would have thought it's DIC. But he said he played around with it quite a bit, and he had to optimize it, and he got a very interesting result here. Yeah? Okay. You're going to make a permanent slide. So I'm going backwards. Yeah, so that's basically, yeah. Can you show microplastics? I think you're answering some of my questions. I hope I did not forget any important questions. Lots of chemicals have changed for safety. No one uses carbon tetrachloride for preserving insects anymore. For preserving insects, I've also tried this a little bit. Of course you can preserve insects by putting them in an insect box, completely dry, or use regular alcohol. So this actually also works. As a matter of fact, I also kept some plant specimens in my in a jar with pure alcohol for now over 20 years. Yeah, so simply because I wanted to see how long you can keep them, and it stores almost indefinitely. Yeah. So yeah, lots of chemicals have changed, and as a matter of fact, people wanted already asking where can you buy Uperol and so on, and you can still buy it. Chemical supplies companies have those because Uperol is used by people who are preparing insects. However, those companies might not sell them to private people. So it's again an issue a little bit. The bottle of Uperol that I've got here, I bought a long time ago from a specialist shop that are basically selling material for entomologists, which are insect collectors. And that's where I was able to get it. However, if you just want to try it out for hobby purposes, I do recommend that you try out the Elmer's PVA glue, okay, the clear one, and dilute it down with a little bit of water. Okay, it might in most cases also work, but again, it depends a lot on the refractive index here. Yeah. Does anyone know if we can upload a picture or a video in a live chat? Unfortunately, it does not seem to work. I've tried this myself a little bit. So what I suggest is the following. If you want to share pictures, then please send them to me at oliver@microbehunter.com. Oliver, then at microbehunter.com, and then I can include, if you send them to me and then you give me permission, and then I can show them to you in a live stream. Some time ago I actually made people sent me pictures to show their microscopy setup, and I kind of showed this as well. But if you have any pictures to share, please do so. Okay, please send them to Oliver at microbehunter.com, and then you give me this way. I won't use them otherwise. I'm just going to then show them in the live stream, and it would be also a nice possibility. Yeah, bioethanol is probably the best, cheapest way to store insects. Yes, alcohol in drugstores is expensive. The reason why some alcohol is extremely expensive is because of alcohol tax. Usually the alcohol that you can buy in hardware stores, usually for cleaning purposes, they contain an additive so that you cannot drink it. It's bitter. And then there's also no alcohol tax on it, and therefore it can be used for technical and for cleaning purposes, and that's actually also what I'm using here. So it's pretty cheap. If you now buy pure alcohol in a drugstore, which is absolutely not necessary, then it can be very expensive because, of course, they want to restrict this as well because it's alcohol, yeah, and it can be consumed theoretically, yeah. So oblique illumination is one of my favorite techniques. A shame no one in college knew how to do it. Many years were missed out. I did a little bit of reading concerning oblique illumination, and I heard that back in the day, in the 18th and 19th century, oblique illumination apparently was extremely popular. Yeah, so this was actually very popular, apparently. I just read this somewhere, and then it somehow got a little bit forgotten. But what I recommend is not only to use oblique illumination, but try to combine it with Reinberg by giving it a little bit of a color as well. Then you're getting also very nice effect. Yeah. So yeah, I think that's, yeah. So over here, again, that's basically the. Also notable, isopropyl alcohol works for some specimens, but most insects will lose their color when stored in isopropyl alcohol. Interesting, I didn't know that, but sometimes it's actually good if insects lose their color, if they become bleached, because sometimes the exoskeleton is simply too dark. So this could also be an advantage, and over here I see another diatom. Yeah. Over here. Actually several of them here. That's a diatom shell. But honestly, I have had nicer specimens before. There are some diatoms which are parallel, maybe this one here as well. Bacteria, they're called. But all of the others here, these are not diatoms. Here again, some, yeah. It's kind of interesting. I'm doing a live stream on diatoms, but sometimes the specimens that I had before showed diatoms much more nicely than what I've got today. But as a matter of fact, mainly I also want to show you a little bit of how to prepare the slides and how to use the centrifuge and so on. Yeah, it's not so many diatoms today. Well, Actually, we do have a lot of diatoms, but they don't look so nice, honestly. That's the thing. Because the nice one in my thumbnail that you've seen, it was actually a huge one. Yeah? And the ones that we see today are actually mostly relatively small. Okay. So what this basically means that I think I have to try out a little bit purifying them and concentrating them, and that might be something like over here, for example, is another one. Yeah. And yep. But before I leave, because somebody actually mentioned this, something so-called forams for foraminifera. Let me see where I found those, because they really look nice. Key number 14. Forams are actually the shells of certain amoeba. Just let me have a look here. Number 3. No, these are radiolarians. Okay, so these are radiolarians. I just want to show them to you because they also look very nice. Again, these are not diatoms, but the so-called radiolarians. I just want to show them to you, a commercial slide here, because they look very nice. Look here, we already got the first ones. And let's zoom in a little bit here. Yeah, they have quite a complex shape. Again, this is a commercial sample here. Yeah, and you have to focus a little bit, because they are quite thick. They have also different shapes here, but some of them have this, quite interesting shape here. So these are, so-called radiolarians. Yeah, also. Yeah. No, they're not alive. Somebody's asking if they're alive. This is a commercial slide. Which basically has also been, I don't know, maybe 20 or 30 years old. Yeah. And you can also find them in sand. Yeah. Yeah. So just also wanted to show you that there are plenty of things. So if you have sand samples, then I would recommend that you also put them under the microscope, and then you might also be able to find those. Yeah. Yeah. Okay, well, I'll keep on looking, and maybe next week I'm going to show you some more. I've got a couple of more samples over there now, but I think because the one in a couple of months ago, I actually took some sediment from a local small stream, a very tiny river, and there were some very large and beautiful diatoms in there, but not a lot of them. Yeah, so I had to look through them, and then occasionally found quite a few large ones. Yeah, they live in sand or underwater. Those radiolarians are basically, they settle to the ground in the sand of marine environments. Yeah. Yeah. So so this is basically the white cliffs of Dover are billions of foraminiferans. Basically they're calcium carbonate shells. So basically this is a completely different material than the diatoms that I showed you. So calcium carbonate is the same material like our bones is made of. Yeah, and like the same material like seashells, for example, and so on. Yeah, this is here. These are some occasional fragments that you find here. But let's go down a little bit with the magnification again. Yeah. So you see that there are plenty of different sizes and shapes here as well. Yeah. Let's have a look at this one here. And this is what I mean. Honestly, I mean, for the microscopy community, that would be actually also something I've been thinking about. I don't know quite how to implement it, but I mean, if around the world, if you have access to, I don't know, to forums or to diatom shells or so, simply because there are many of you in your area, I've been actually thinking about starting some kind of like an exchange, how do you call this? Exchange platform for microscope slides, where you can essentially make your own microscope slides and you kind of trade them. Yeah, that would be kind of nice. So because, but how would you implement something like that? Yeah. And then people could actually make their own microscope slides and I don't know, yeah, permanent mounts, and you kind of trade them in, you offer them, and then you basically get in return something else. I think it would be a nice idea, but yeah, yeah. Okay, folks, I've been talking again long, 1 hour, 40 minutes already. Yeah, if you have any requests or any ideas, then please do mention to comment on those. Yeah, can you make a video about how to advance oblique illumination techniques like you made a beginner's guide? Could you make it a pro version of it? Okay, if I do something, yes, I could do that. I will do that. I've been thinking about this, but I will not do this in the form of a live stream because my microscope that I have right now does not allow me to do that. But I've got another microscope, and I will actually probably yeah, experiment more with that. Okay, because yeah, it's not so difficult, and indeed combining oblique illumination and also some color filters is quite nice. Yeah, yeah. Okay. Yeah, I think I'm just going to leave it at that. Again, wish you all the best. I just want to make a short announcement that I will not be doing a live stream on the 10th of June because I'm not around. I have an appointment at that time, but I might move it maybe for a day or two, or for a couple of, I don't know. Yeah, but on Saturday, the 10th of June, I might not be able to do a live stream. Just want to announce it, but I might catch up on this, yeah, a little bit on a different day. Okay, yeah, I'm going to leave it at that. I hope that it was interesting again, and yeah, last comment. I looked at the water from a puddle nearby a river and found some microscopic crustaceans, Daphnia and protists. Haha, yes, and as a matter of fact, my video, the next video, which I might upload tomorrow or so, today or tomorrow, is all about some Daphnia and water crustaceans that are found in a puddle in a forest. Okay, so yeah, I wish you all the best. Yeah, enjoy microscopy, happy microbe hunting as always, and see you next week again. Okay, bye bye, all the best.